Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to talk about sickling or sickle cell test. We can also call that a sickle cell disease test or sickle cell anemia test as well. Now my name is Muhammad Kamil Khan and you guys watching Kami microbiologist channel. Let's start the video. First of all, let's talk the main content, means what we are going to talk about in this video. In this video we are going to see that what is actually sickle cell disease and what are actually sickle cell test, what are sickle cell test means. Then we are going to see the purpose, the importance or the uses of this test, means why we perform that test. Then we are going to see the principle or the material, the things which we used for that test, what are the purpose of that material or the things or reagents. Then we are going to see the requirements, means what we need while performing that test. Then we are going to see that how many methods we have while performing that test. After that we are going to see the procedure, means how we can perform the test. Then we are going to see the results of that test. So first of all, let's start with the introduction. First of all, what is actually the sickle cell disease test means? The sickle cell test or sickling test is actually again the hematology or the blood test. All of you better know that this is again the hematology test videos lecture. And in different hematology test videos lecture, what we talked different types of the hematology test like CBC, we talked ESR, and we talked HB electrophoresis as well. So again, this is the video lecture that is related to the hematology test. And we have the sickle cell test. So sickle cell test is actually the test in which we mostly check the sickle cell disease like anemia, sickle cell anemia, or we are mostly see the normal hemoglobin concentration in the blood. Then we have the sickle cell disease. Remember guys, sickle cell disease, in the sickle cell disease what we have the RBC disorder, in which the RBC are become in a sickle shape, means the RBCs look like in a rupture form or C like shape RBC have. Now then we have the most important thing, that remember guys that sickle cell disease is actually the genetic disease. There is a mutation in the RBC cells due to which the RBC are become into the C shape, and this type of disease can also be transferred from parents to the offspring. Then we have some important things, that remember guys, we have to perform that test in case of newborn baby, and we also perform that type of the test in case of older or adult children when it is needed. Let's suppose if they have anemia like symptoms, means they have fatigue, they have headache, they have chest pain, their skin are become pale yellow, we can say like that. So it means that they have anemia. After that we perform that type of the test in case of the adult or in children as well. Now in the picture, as you can see in the sickle, in this picture you can see that the normal size and shape of the RBC. So these are the normal shape of the RBC, as you can see. So all of you better know RBC are the biconcave or disc like shape, as you can see. Now these are the normal RBC, and these are, as you can see, these are the abnormal RBC. So as you can see, these are just like a C in shape or rupture in form, as you can see. This is called sickle cells, sickle shape of the cells. So when you found under the microscope, when you found these types of the cell, so it means that the patient have sickle cell or sickling disease, the patient have sickle cell disease or sickle cell anemia, we can also say like that. Now what we required while performing that test? In this test we need blood in EDTA tube, as you can see. So we need blood in EDTA tube. We need sodium, 2% sodium metabisulfite, as you can see. mostly the sodium metabisulfite are in a powder form, so we put the powder into the distilled water and then we will mix it, so we get sodium metabisulfate solution. We need glass slides, as you can see. We need cover slip, so these are the clean glass slide and cover slip. We need wax or we need Vaseline. Okay, we need a micro test tube, as you can see, microscope, and we also need Atlas reagents. So these are actually Atlas kits, and we mostly use these Atlas kits when we perform sickle solubility test, because it contains reagent 1 and reagent 2. The reagent 1 is actually solution in form or in a liquid in form, and the reagent 2 are in powder form. Then we need jester, as you can see, or we also call that pipette, and these are the most important material or the things which we need while performing that test. Now let's see the principle. So remember guys, in this case we mostly use the Atlas kits, means reagent 1 and reagent 2, and we also used sodium metabisulfite. What are the reason behind that, why we use this? Actually, whenever we use this, so it can mostly reduce the oxygen tension. Remember guys, it reduces the oxygen tension. Now once the oxygen tension is reduced, what happen? As a result, we get sickle shaped RBC. That's why it is really important to use. Then we have methods. So how many methods we have? We have almost 2 methods. The first method is called simple slide method, as you can see. So in this case we need a slide with a cover slip, and then we put our blood sample as well as sodium metabisulfite reagent, and we will mix it, and then we make the slide, and then we see it under the microscope. We'll talk it in the next slide. And we also need then we have another method which we call it sickling solubility test method. So in this case we need the test tube, and so as you can see, this is the sickling solubility test. First of all, let's talk about that how we can use the slide and performing the metabisulfite method to determine the sickle disease. First of all, what we have to do, we just take 1 drop of the blood from the EDTA tube and we'll mix it with the 1 drop of the sodium metabisulfite solution onto the clean and dry glass slide. Once we do that, after that what we do, we have to put a cover slip. Now once the cover slip is put it, then it should be sealed, means their edges should be sealed using wax or Vaseline. Then we let the slide for almost 1 to 4 hours at room temperature, and after that what we do, we have to visualize the slide under the light microscope. Almost means mostly at 10x we will focus the slide, and at 40x we visualize all the cells. Mostly in this case we visualize the sickle shaped cells, sickle shaped RBC cells. As you can see, this is the slide under the microscope. So under the microscope you will find the structure like that. So these are the normal RBC, as you can see, these are the normal, and these are the abnormal RBC. So C-shaped, like C-shaped cells, as you can see. This is called sickle cell, as you can see, sickle shape. Okay, so you have to count these cells in using 10 field. When you count it using 10 field, and if you found a large number of the sickle shaped cells, so it means it is the positive case, and if you found a less number, so it means that it is the sickle cell trait. So this is how we can perform that test. Second, we have another method which we call it sickle solubility test method. Now how we can perform that? First of all, we have to mix the reagent 1, which is actually powder form, and reagent 2, which is actually liquid form. Once we take it, so then what we will do, we have to mix it like in 8 shape. So let's suppose this is the reagent, so we have to mix it like that into 8 shape. Okay, once we mix it, then what we will do, we have to take the 3 clean and dry glass test tube and we'll label it. For example, the first test tube will be labeled positive, second test tube or the first test tube will be labeled negative, second test tube will be positive, and the third test tube will be labeled patient sample. Now after that, in the first tube, what we will do, we have to add negative controlled reagent, and in the second tube what we do, we add the 2 ml of mixed reagents, means this one, reagent 1 and reagent 2, which we mixed. So we have to add it into the second and third tube, 2 ml respectively. After that, what we will do, we have to take 20 microliter from the positive controlled reagent, which is actually present in the Atlas kit, and will add it into the second tube. Once we add it, so what we do, we have to shake it gently and will incubate it for 5 minutes. The same method we also do with the third tube, but in this case we add 20 microliter of the patient blood instead of using positive controlled reagent, and then we also shake it and incubate it for the 5 minutes. Now after 5 minute incubation, what we do, we have to compare the 3 test tube with reading chart. This reading chart actually contain lines or squares. Now how we can read it? So let me show. As you can see in this case, the first test tube, as you can see, this is actually negative. Now why this is negative? If you found the lines, as you can see, this is actually the chart or reading chart we have. Now if you found the lines in the test tube, as you can see, it means that there is no turbidity. That's why you can easily visualize the lines. So it means that it is transparent and there is no turbidity. If there is no turbidity, it means that this is negative. So the result is negative. In the second test tube, as you can see, there is turbidity, and due to turbidity you did not visualize the lines that is not been seen in the test tube. As you can see, if there is a less we can say if there is a less lines observed, so it means we have sickle cell trait. But in this case we can say that it is actually sickle cell disease. We can also say like that. Now we have another method which we called it rapid method to find out the sickle cell disease. In this case we need a strip. As you can see, this is called as HB strip. This HB strip contain 4 bands. As you can see, the first band is actually control, second is HbA, means hemoglobin adult, hemoglobin sickle or hemoglobin C, which is actually the thalassemia indication. So remember guys, if we get control bands with control and bands with HbA, it means the report is normal. If we get bands with control, bands with HbA and bands with HbS, less band with HbA and some of the bands with HbS, it means that we have a sickle cell trait. And in this case, if there is no bands in HbA as well as in HbC, but there is band with control and HbS, so it means that this is positive sickle cell disease. As you can see in the fourth picture, as you can see, there is band with control, band with HbA and band with HbC as well. It means that this is actually the thalassemia, we can say like that. In the fifth picture, as you can see, there is a band with control and band with HbS as well as HbC, but no band is observed in case of A. It means that this is positive thalassemia, or we can say thalassemia like disease, positive thalassemia disease. So using these strips, we can, after almost 5 minutes it will give you the report. So after 5 minutes you will watch the bands in this strip. So this is actually the rapid method while performing the sickle test. Now one thing more that I must tell you, remember guys, this is just for the indication that we have sickle cell disease. Using these three using these 3 methods, we just check that whether we have positive result or negative result. Once we got the positive sickle cell disease result, so then what we will do, we have to perform HB electrophoresis, which are really important to know the percentage of HbS, or we can say hemoglobin S, or sickle cell disease, percentage of the sickle cell disease. So we have to perform that test, because in this test you will just see the positive and negative report, but in the HB electrophoresis you may see the percentage of the HbS. So that's why after that HB electrophoresis is also important, and I already explained the HB electrophoresis. You can also check it into my channel. Okay, so that's all about the sickle cell test. I hope you understand, and if you like this video, so please hit the like button, share this video with your friends. If you have any kind of question in your mind, you can write your question into comment. Inshallah, I will try my best to give you answer as soon as possible. Also subscribe to my channel to get more interesting videos like that. Thanks for watching.